Hey guys, I hope you're doing fine. It's been a long time since I'm recording a lecture, and this is the end of the study, guys. This is it. We kind of made it. This is the last lecture in the study, and I hope you've been having enough time to fill the pre and the post quizzes on time. Okay, so let's do this. Today we're going to be discussing the placenta. It would be nice to understand how we've reached till here via the inner cell mass and everything that lies outside here. Well, that's the placenta, and that is derived from the outer cell mass, which eventually forms the trophoblast, and then the placenta. So if you were to define the placenta, you would say it is a discoid shaped 15 to 20 centimeters diameter organ that is responsible for communicating between the mother and the fetus. And it becomes a highly specialized organ as it evolves. So everything that I just described is what it looks like at term. Let's try to see how we begin from the implantation stage to reach to this beautiful looking structure called the placenta. Now, I want you to follow a clockwise scenario here. Then it would be chronological, right? Let us start with day 6. So somewhere over here we are at day 6, and remember we're going to only concentrate on the outer cell mass now. That's in light green over here, okay? Whatever that shade of green is. Now, you can see that there are two shades here. So the outer cells lose their cell membranes, form a multinucleate mass that is known as syncytotrophoblast, dark green, yeah? By the time we reach the 7th to 8th day, you can see that the syncytial mass is increased and is encroaching upon the uterine endometrium. Now, after implantation occurs on day 7, the uterine endometrium is just called the decidua. Okay, that's inspired by the decidual trees concept, if you remember your botany. So, the decidual tissue is maternal, and it comprises of the endometrial fibrous connective tissue stroma. Okay, you can just call it the uterine stroma. It's basically connective tissue cells, and the dark green colored thing is your syncytotrophoblast, and one is encroaching upon the other. What you see around are these little red dots. They are just maternal capillaries which are slightly bursting open. So small pools of blood have already started forming here. What you see in light green is what is known as the cytotrophoblast, which is the layer where the cell membranes haven't disappeared. Okay? Now, by the time you reach the 9th day, look what's happening. Okay, ignore what's happening inside here because that's not our concern. But the syncytotrophoblast has little spaces appearing, right? And these spaces, my friends, are known as lacunae. Lacunae. Okay? And these lacunae eventually form the intervillus spaces. At the same time, the sinusoids are getting bigger and bigger. They're labeled here as enlarged blood vessels, right? And there are certain changes in the surrounding connective tissue stroma as well. And these changes are nothing but an increased glycogen deposition in these cells. And that glycogen is going to be converted into glucose and used as fuel, right? So this reaction where the connective tissue stroma cells are being laden with glycogen and enlarging is known as the decidual reaction. What is it known as? The decidual reaction. Okay? And then we reach the next stage, that is the 12-day stage. Now, in the 12th-day stage, the red has finally combined with the lacunae. So the lacunae are now filled with maternal blood, and this is like the earliest signs of an establishment of uteroplacental circulation. You can see that happening here in real time. a maternal sinusoid opening up into the intervillus space. Uteroplacental circulation. Now, if you observe carefully, the cytotrophoblast is slightly becoming taller in certain regions and shorter in certain regions. These tall regions are eventually going to grow taller and form pillars. And these pillars, my friends, are known as chorionic villi. Okay? Before that, I also want you to know that the extra embryonic mesoderm has just been introduced in the embryoblast. And this, in further stages, is also going to encroach into the cytotrophoblast. Right? So let us understand what exactly is a chorionic villus. This structure over here would be an example of one chorionic villus. Okay, villus means a finger-like projection. So we're at day 13. The lacunae are becoming bigger. The syncytotrophoblast is becoming thinner, and the cytotrophoblast is emerging up as pillars. Now, if you observe carefully, the extra embryonic mesoderm is also forming the core of these pillars. Can you see that? It's happening in this area, it's happening in this area as well. So, by the time we reach the end of the third week, somewhere around day 21, look at what the structure is looking like. So now you've got an outer dark green portion on either side of which is the red intervillus space. The lacunae can just be known as intervillus spaces now, and they're filled with maternal blood. So you've got an outer dark green layer. Inside that you've got a light green layer of cytotrophoblast, and finally you've got this orangish pink layer of extra embryonic mesoderm. To make things more complicated, can you see that there's a slight dark red region here? Well, that dark red region is the development of capillaries, or other fetal capillaries, within the extra embryonic core. So now you have syncytotrophoblast outside. Just within that you have the cytotrophoblast. Within that you have the extra embryonic mesoderm, within which is developing the fetal capillaries. So according to this, the chorionic villi are differentiated into primary, secondary, and tertiary. So let's have a closer look at what that looks like in a transverse section. Now that's a primary villus, outer syncytotrophoblast, and it has a core of cytotrophoblast. Okay. Next is the secondary chorionic villus, where the extra embryonic mesoderm is the newly added layer, right? So you've got the syncytotrophoblast out, within that the cytotrophoblast, and within that the extra embryonic membrane. This structure is known as a secondary chorionic villus. And then as we reach day 21, can you see these little things? These are your fetal capillaries, and they are basically developed within the extra embryonic mesoderm by the cells of the extra embryonic mesoderm itself. A process known as angiogenesis, which you don't need to get into the details of, but the tertiary chorionic villus from outside within consists of syncytotrophoblast, cytotrophoblast, extra embryonic mesoderm, and finally fetal capillaries, or you would probably call it fetal endothelium. Fetal endothelium. So now, from outwards within, or the best way to say it is from the maternal to the fetal side, you have the syncytotrophoblast, then the cytotrophoblast. Please bear in mind that the cytotrophoblast is situated on a basement membrane. Okay? Then the extra embryonic mesoderm. Then the fetal endothelium, which will also be having a basement membrane first. Then the fetal endothelial cell itself. And finally the fetal blood. So that is your early placental barrier. And you wouldn't use the word membrane here, because there's a lot of layers, right? So the early placental barrier is formed from outside within, or from the maternal to the fetal side by the syncytotrophoblast, the cytotrophoblast, then the basement of the cytotrophoblast, then the extraembryonic mesoderm, then the basement of the fetal endothelium, then the fetal endothelial cell itself, and then the fetal blood. Okay, so that's kind of what they've shown over here. Check it out. Now this upper diagram shows the primary villus, that is around 7 to 8 days. The secondary villus, that is around 12 days. The extraembryonic mesoderm core has now arisen, and the 21st day where the fetal capillaries have started. And what you see over here is a picture at the end of the 4th week of development, where you can see that the maternal vessels are penetrating inside and pouring in blood into the intervillus spaces or the lacunae. And on the fetal side, the fetal capillary system is nicely developed, right? Can you see that? And this orangish red thing is the extraembryonic mesoderm, which eventually forms what is known as the chorionic plate. Okay, so by now I think you are starting to understand that the placenta has a dual origin, where the fetal part of the placenta is developed from the chorionic plate, which is nothing but the extraembryonic mesoderm, and the maternal part is contributed by what is known as the decidua. That is all this here. Okay, so we're going to see what exactly happens to the decidua and what is the further development of the chorionic plate in the next slide. Now this is the end of 4th week over here, then the 4th month directly. So now we're taking a big leap from the 4th leap to the 4th month. So let's discuss the changes that take place. Okay, so change number 1 is the development of a cytotrophoblast shell. So technically, the cytotrophoblast that is present internally and within the syncytotrophoblastic layer extrudes out and eventually goes on either direction to form a shell that is known as the cytotrophoblastic shell. Kindly note that whenever the maternal blood is going to come in touch with the fetal tissue, there will always be a layer of syncytotrophoblast present. You can see that here in dark green. Can you see that? Okay. Now, the second change that takes place is the development of what is known as spiral arteries, spiral vessels essentially. That's basically a maternal vessel that has a slight spiral appearance, and a very interesting thing that happens over here is that the cytotrophoblast erodes into these maternal vessels and forms a new hybrid kind of a vessel. So the cytotrophoblast undergoes what is known as epithelial to endothelial transition. Okay, and you can use a fancy word like endovascular invasion by the cytotrophoblast. What happens after this invasion and after this conversion is that the maternal vessels, or the so-called spiral vessels, are converted from a high resistance low diameter vessel to a low resistance high diameter vessel. That's just fancy terms for more blood starts pouring out of these as compared to before. Okay, now by the time you reach here, you can see that there are villi that extend all the way on either side, that is from the chorionic plate to the maternal region, which is known as the decidual plate. Okay, so these villi that extend on either sides are known as anchoring villi. Okay, they are known as anchoring villi, and the anchoring villi eventually lead to these sprouting villi, which are known as free villi. Okay, and they are further named as ramus and ramuli, the branches of the branches. So it's essentially a villus tree. As you can see, this could be like the main trunk, and these are all the branches, and then it's got all these little smaller branches, right? Very much like a tree. And this villus tree essentially increases the surface area available for exchange by many fold. Okay? Finally, what happens is the thinning out of the placental barrier. So you can see what the barrier looks like over here in the 4th week, and it constitutes of these 4 layers: syncytium, the cytotrophoblast, its basement membrane, the connective tissue, and the basement membrane of endothelium, and then the endothelium. But by the time you reach the 4th month, you reach the 2nd half of pregnancy, most of these disappear. The cytotrophoblast eventually thins out, the connective tissue eventually thins out, and the placental barrier, or let's call it the placental membrane at term, is formed basically just by syncytium and endothelium. Again, whenever you say endothelium, always remember that the basement membrane is what comes first, and then the endothelial cell. Okay, so this is what happens in the 2nd half of the pregnancy. The vessels are now known as spiral vessels. There is an epithelium to endothelium transition, because of which more blood is now flowing out. Thus, there is more blood that is reaching the fetus. The villi are arborizing. New branches are coming, known as ramus and ramuli. And finally, the placental barrier is extremely thinned out for better exchange. So as you can see, as the placenta is evolving, the fetus is also growing, and the fetus is going to need more blood, and that is taken care of by all these little factors. Okay, now let's see what exactly is the chorion, the amnion, and the decidua. So let's zoom out as much as we can to reach a stage over here. Now this is the end of the 2nd month in A, and here we've got the maternal portion of the future placenta, which is known as the decidual plate, right? So before it even forms a plate, the region where the placenta is going to well develop is known as Decidua Basalis. Okay, this is the region of the future placenta. Okay, then the part of the decidua that is surrounding the developing embryo is known as the Decidua capsularis, and the remaining decidua that is just lining the uterine cavity is known as the Decidua parietalis. So this is the 3 types of decidua that you need to know. Now, the trophoblast, or now we just use the word chorion, which is nothing but the fetal part of the placenta, that is well developed and extending to form all these villi, is known as the chorion frondosum, which you can see here, right? And the part that is not very well developed, not having multiple villi and just slightly flattened, is known as the chorion levi. So these are the 2 types of the chorion that you need to know about. Okay? Now see what happens as the embryo grows. Okay? You can see that the chorion frondosum has developed along with the Decidua basalis to form the placenta. That's your connecting stalk, which is now formed the umbilical cord. Okay? So the placenta is contributed by the chorion frondosum and the Decidua basalis. This is the decidual plate. This is the chorionic plate. Okay? And you can also see that the uterine cavity is completely obliterated, which means that the Decidua capsularis fuses with the Decidua parietalis. That's your fused Decidua parietalis, chorion levi, and amnion. So this cavity is the amniotic cavity. This cavity is the chorionic cavity. Eventually, the chorionic cavity also completely obliterates as the amniotic cavity enlarges. So you have the fused amniochorionic membrane. Okay, that's your amniochorionic membrane. Alright, so once again, let's have a look at what exactly happened. The Decidua parietalis fuses with the Decidua capsularis, and the uterine cavity is completely obliterated. Then the amniotic cavity enlarges and completely obliterates the chorionic cavity to form the fused amniochorionic membrane. Now this is the one that is essentially popping out, and puncturing this is what exactly happens when the water breaks. That's nothing but the spilling of the amniotic fluid that is present over here. Okay, so I hope we understand what is chorion, what is amnion, and what is decidua. Let's have a look at the placenta in the second half of pregnancy. As you can see, it's looking like a very beautiful structure now. This is the decidual region, and the decidual region starts going down, forming what are known as decidual septa, but they never quite make it the chorionic plate. So they do not completely cross and meet the other end like the anchoring villi. Okay, you can see the spiral arteries here. Now the spiral arteries usually open in the center, if you view from the maternal side, the center of the cotyledon, and blood is gushed inside, and then centrifugally, once the blood starts going towards the periphery, it is collected by the spiral veins or the decidual veins, which you can see in the periphery over here. Okay, so that kind of happens from a center towards the periphery thing. Yeah, let's have a look at the chorionic plate. This is the amnion that is fused with the chorion. This is the umbilical vessels. This is the umbilical cord, by the way. And these are the umbilical vessels that are developing in the umbilical cord. We've got 2 umbilical arteries and 1 umbilical vein. More on that later. So these are the umbilical vessels, and you can see that branches from the umbilical vessels are basically going to contribute here to form the fetal capillaries, right? So now you've got the maternal side coming from here, you've got the fetal side coming from here, and I've already discussed about the placental barrier in detail, so I won't talk about that again. Okay, let's look at what the placenta appears like at term or at birth. So at term, the placenta is discoid in shape, about 15 to 20 centimeters in diameter and about 3 centimeters thick. It's about 500 to 600 grams in weight, and it is expelled about 30 minutes after birth. Okay, and this is what it looks like. It basically has the side that consists of the fused amniochorionic membrane. So you can see the amnion here, you can see the chorion here, or the chorionic plate, and all these little radial vessels are essentially the chorionic vessels, and all of them kind of join together to form the umbilical vessels, which are a constituent of the umbilical cord. Now the umbilical cord may not always be attached to the center of the placenta. It could be somewhere over here marginally, that's known as marginal placenta. There are multiple other types of abnormal insertions, but I want you to read about them on your own. Okay, so what you're staring at here is essentially the fetal side. The maternal side looks something like this. It has multiple structures known as cotyledons. Can you see these little grooves in between cotyledons? These grooves are the decidual septa that go deeper down but do not make it all the way to the chorionic plate. Okay, and what you see over here, the outer lining is essentially the Decidua Basalis. Another important thing is the placental circulation. So at term, the placental circulation has greatly increased. About 150 ml of blood traverses within the intervillous spaces at a point of time, and this is replaced about 4 to 5 times per minute. As I already explained, the Villus tree increases the surface area to a large extent, the total surface area being about 4 to 14 meters square, and there is a great exchange of material through this extensive surface area. Also remember that the human placenta is known as hemochorial because the hemo, or the maternal blood, is in intimate contact with the chorionic plate, that is essentially the extraembryonic mesoderm connective tissue. Okay, now let's talk a little bit about the functions of the placenta. I'm not going to go into the details of these functions, just read about them, but essentially they are exchange of gases. Now that's no rocket science. Oxygen and carbon dioxide. Then there is obviously exchange of nutrients and electrolytes. So the nutrients from the mother reach the fetus, and also the waste products of the fetus reach back to the mother through the placenta. Okay. Then a very important function is immunity. Maternal antibodies of the IgG type, and you don't need to know the details about that, but that's the most important antibody that can cross the placental barrier. The IgG type antibodies, they cross from the mother to the fetus. Do note that though the maternal antibodies can prevent most diseases, infections, certain viral infections cannot be prevented by these antibodies. So there are certain viral infections that can be transmitted from mother to child. Okay, more details on that in a separate section somewhere else. Lastly, hormones. Now the placenta is responsible for secreting many hormones. Some of them include HCG, or the human chorionic gonadotropin. Now it is this hormone that is initially secreted by this inside trophoblast and then continued secreting by the placenta. Point number two is progesterone, and initially progesterone is done by the corpus luteum, and later this function is taken over by the placenta. By about the beginning of the fourth month, it is the placenta that secretes the progesterone and maintains pregnancy. Another important hormone, I'm going to write it here, is what is known as somatomammotropin. Okay, again, we don't want to talk a lot about these right now, but just remember somatomammotropin and also estrogen. So some of these hormones are secreted by the placenta. Okay. Before we summarize everything, I just wanted to talk a little bit about the umbilical cord. So you can see that over here. The connecting stalk eventually develops into what is known as the primitive umbilical ring. And in the early stages, we have a stalk that connects the developing gut tube to the yolk sac, and this stalk is known as the vitello-intestinal duct. We've spoken about this before. Okay, so this vitello-intestinal duct is one of the early contents of the primary umbilical cord, but eventually the intestine rolls back inside. This is known as physiological umbilical hernia, again for another day. Then we have a couple of other things like the extra embryonic coelom or the chorionic cavity, which also extends into the early umbilical cord. Eventually, that is completely obliterated when the chorion and the amnion fuse. Okay, so that is also not really a content. So at term, the only things that are present in the umbilical cord are the umbilical vessels, which are the two umbilical arteries and one umbilical vein. Okay, we'll talk about where the other vein disappears in systemic embryology at some point of time. And the connective tissue in between these is known as Wharton's jelly. Okay, it's like mucoid connective tissue. Again, it just acts as a protective covering for the umbilical vessels. All right. So at term, the contents of the umbilical cord are the two umbilical arteries, the umbilical vein, and the Wharton's jelly. Everything else that I discussed before is something that you don't need to know about in detail right now, but they are a part of the primitive umbilical cord. To summarize the entire lecture, let's have a little bit of a time zone thing happening. So around the 8th day, the trophoblast differentiates into cytones inside a trophoblast. The lacunae appear on the 9th day. Then the primary villi start forming at 13th day. Secondary and tertiary villi start forming in 16th day. Please call them chorionic villi just to be more precise. By second month, villi are seen all around the trophoblast, but more so towards the future placenta, which is known as the frondosum. And a definitive placenta is formed at fourth month, and at full term it is you've seen what it looks like, but this is a beautiful picture from Langer's. This is what it looks like. This is a full term placenta connected by the umbilical cord. Okay, this is the fetus inside the uterus, and you can see the placenta at this region, right? So we just went the other way around. This is full term. This is also around 18 weeks. And this is something as early as the 6th week, where you can see the developing embryo, and you can see the well-developed chorionic frondosome on this side, and chorion leve, the lesser developed chorion leve on this side, frondosome on either side here, and you can see the primitive umbilical cord connecting. Okay, so thank you so much. With this, I announce the end of the general embryology series, but we've got more things coming up. So stay in touch, and if you have any questions about this lecture, because there's so many things to cover and I could not cover all of them, you can always leave them in the comment section. All right, so I'll see you with the next series, and it's mostly going to be abdomen, so that's going to be fun. Bye bye.